Hello friends and welcome. Today we shall talk about food spoilage enzymes PPO, that's polyphenol oxidase, and cell wall degradation enzymes. The main objectives of today's deliberation are: first, to understand the concept of food spoilage, followed by underlining various factors of food spoilage. Then we shall highlight the chemical changes that cause food spoilage and also try to understand the concept of enzymatic browning. Further, we shall try to get a general idea about cell wall. And finally, we shall try to get a thorough idea about cell wall degrading enzymes. To begin with, let me first of all give you a brief introduction about food spoilage. Every food item that we eat is biological in origin. That's, it comes from living organisms. Thus, it's bound to contain proteins, carbohydrates, and fats. And several metabolic activities would be carried out in them with the help of enzymes. These carbohydrates, fats, and proteins are a source of energy. Since every living organism requires energy for survival, we consume food for procuring energy, and similarly, even microorganisms require energy. Thus, food becomes the target for the growth of some microorganisms. Food spoilage means the original nutritional value, texture, flavor of the food are damaged. The food becomes harmful to people and unsuitable to consume. Having understood the basic introduction about food spoilage, let's move on to the factors of food spoilage. Nutrients in food, their kind and proportions determine the type of organism that will grow. Also, microorganisms vary in their ability to use nutrients. The presence of easily utilizable nutrients will encourage faster growth and quicker damage. For example, a food with easily utilizable sugars will allow better growth than one which contains polysaccharides. Most foods contain enough peptides and amino acids that they can meet the nitrogen requirement of most organisms found in foods. Some organisms are also proteolytic and can grow on proteins found in the food. The mineral requirement of microorganisms is generally met by the food, and this is not a limiting factor. Some foods may contain antibacterial substances which may prevent bacterial growth and food spoilage. Other than the nutrients present in food, factors such as the pH, moisture content, oxidation reduction potential, etc. also influence microbial activity in foods. The oxygen tension or partial pressure of oxygen and the reducing and oxidizing power of the food, that's oxidation reduction potential, it influences the growth of organisms. In relation to oxygen, bacteria can be aerobic, anaerobic, or facultative, while fungi are mostly aerobic. Yeast are aerobic or facultatively anaerobic. A high oxidation reduction potential favors the growth of aerobic and facultative organisms. Most fresh animal and plant foods have a low oxidation reduction potential in their interior, but have a higher oxidation reduction potential outside. Thus, a fresh piece of meat could support the growth of aerobic organisms in the exterior and the growth of anaerobic organisms inside. Other than microbial growth and destruction, food is spoiled by enzymes present within them. This self-destruction is termed as autolysis. The most important mode of destruction is by enzymes. Enzymes are proteins found in all plants and animals. If uncooked foods are not used while fresh, enzymes cause undesirable change in color, texture and flavor. This is because even though the vegetables and fruits have been plucked from the plants, their cells are still alive and continue the basic life process, that's respiration. Now we will discuss another segment of this topic, that's chemical changes causing food spoilage. Food spoilage occurs in food products due to reaction or breakdown of the chemical components of the food, including its proteins, lipids, and carbohydrates. The rate at which the chemical reactions take place depends on many factors, which are water activity, temperature, pH, exposure to light or oxygen. Protein degradation can involve reactions with protein and other ingredients brought about by enzymatic activity. Protein hydrolysis is achieved by enzymes collectively called proteases. Petrification is also protein degradation, where decomposition of animal proteins is carried out especially by anaerobic microorganisms, described as petrifying bacteria. Petrification usually results in the formation of amines such as putrescine and cadverine. Atmospheric oxygen reacts with food components and causes rancidity. Rancidity is the term used for the deteriorative changes of fat with time, and it results in undesirable flavor and odor. Hydrolytic rancidity occurs in foods when the lipid, that's fat, is hydrolyzed by the water contained in food to fatty acids. Some of the liberated fatty acids are volatile and some have very unpleasant odors and flavors. When rancidity occurs due to air, it's termed as oxidative rancidity. The oxidation of acyl glycerols, which occurs in air without the presence of enzymes, is called autooxidation. Among the products of autooxidation are hydroperoxides, that's ROOH. These have no taste, but they decompose easily to form aldehydes, ketones and acids, which give oxidized fats and oils their rancid flavors. It can be slowed down by addition of antioxidants. Now we shall discuss an important phenomena, that's enzymatic browning. Polyphenol oxidase, that's PPO, is a generic term for the group of enzymes that catalyze the oxidation of phenolic compounds to produce brown color on cut surfaces of fruits and vegetables. The action of PPO leads to major economic losses in some fresh fruits and vegetables, such as Irish potatoes, lettuce, and some other leafy vegetables. Apples, apricots, bananas, grapes, peaches, and strawberries. In some tropical fresh fruits, up to 50% can be lost due to enzyme-caused browning. Browning also leads to off flavors and losses in nutritional quality. Therefore, the consumer will not select fruits and vegetables that have undergone browning. Black spots in shrimp are caused by PPO-catalyzed browning. The browned shrimp are not acceptable to the consumer, and they are downgraded in quality. PPO activity in plants is desirable in processing of prunes, black raisins, black figs, zapote, tea, coffee, and cocoa. And it probably protects plants against attack by insects and microorganisms. Subsequent investigations showed that substrates for enzyme are oxygen and certain phenols that are hydroxylated in the ortho position adjacent to an existing OH group, further oxidized to orthobenzoquinones and then non-enzymatically to melanins. Millions of dollars are spent each year on attempts to control PPO oxidation. To date, none of the control methods are entirely successful. Moving on, let's shift our focus to chemistry of enzymatic browning. Control of enzymatic browning in fruits, vegetables, juices and wines requires chemical knowledge of the types of phenolic substrates present in a particular plant. The level of reducing compounds such as ascorbic acid and sulfhydryl compounds, the level of oxygen accessibility, nature of co-oxidizable compounds present, and the pathways of polymerization and degradation of the orthobenzoquinones. It's also essential to understand the level of PPO and substrates available at different stages of plant development. Above all, it's important to distinguish between enzyme-caused browning and non-enzyme-caused browning, that's the Maillard reaction in foods. Some PPOs hydroxylate monophenols to give orthodihydroxyphenols, which are then further oxidized enzymatically to orthobenzoquinones. The yellowish orthobenzoquinones are very reactive and unstable. Further non-enzymatic reactions with oxygen lead to additional reactions to give complex products such as indole 5,6-quinone from tyrosine. For example, with further polymerization to melanin and reaction with nucleophiles such as amino groups of proteins. The orthobenzoquinones can react covalently with other phenolic compounds by Michael addition to give intensely colored products that range from yellow, red, blue, green and black. Orthobenzoquinones also react with aromatic amines and thiol compounds, including those in proteins, to give a great variety of products, including higher molecular weight protein polymers. Having understood the concept of enzymatic browning, now let's shift our focus to inhibition of enzymatic browning. In theory, PPO catalyzed browning of fruits and vegetables can be prevented by heat inactivation of the enzyme, exclusion or removal of one or both of the substrates, that's oxygen and phenols, lowering the pH to 2 or more units below the pH optimum, by reaction inactivation of the enzyme or by adding compounds that inhibit PPO or prevent melanin formation. Hundreds of compounds have been tested as inhibitors of enzymatic browning. Exclusion and separation of oxygen and phenols from PPO prevent browning of intact tissues. Commercial utilization of these methods are being examined by numerous researchers. Fruits and vegetables have skins, that's waxes and other surface layers, that exclude oxygen as long as there is no damage to the skins. PPO is physically compartmentalized from phenols in the intact cell. Commercially, oxygen can be excluded from or reduced in concentration in fruits and vegetables by controlled atmospheric storage, packaging techniques, etc. Phenols can be removed from fruit and vegetable juices by cyclodextrins or by treatment of cut surfaces with oxygen impermeable coatings. PPO activity can be decreased by modifying the pH. The pH optima of most PPOs are near 6, although there are some exceptions. Reducing compounds such as ascorbate, sodium bisulfite and thiol compounds decrease browning by reducing the orthobenzoquinones back to orthodihydroxyphenols, or by irreversible inactivation of PPO. Maltol does not inhibit PPO, but it prevents browning by its ability to conjugate with orthobenzoquinones, while kojic acid is effective in preventing browning by both reacting with PPO and with orthobenzoquinones. Competitive inhibitors such as benzoic acid and 4-hexyl resorcinol are useful in controlling browning in some food products. 4-hexyl resorcinol is a very good inhibitor of enzymatic browning of shrimp. apples and Irish potatoes. Now let's highlight the phenomenon of cell wall degrading enzymes. Plant cell walls consist primarily of polysaccharides, the most abundant of which are the cellulose and pectic materials. The hemicelluloses too constitute a sizable portion of the plant cell wall. Cellulose also has the importance of being the most abundant organic matter on earth, whose degradation is indispensable for the recycling of elements in the biosphere. Microbial degradation of cellulose thus is of paramount importance. Efforts are underway to obtain better and more active cellulolytic microbes by discovering them in nature. Developing mutants and the new possibility of genetically engineered organisms for hyperenzyme production. Plant pathologists have since the beginning of this century looked into the production of plant cell wall degrading enzymes, as these are indispensable for making growth in host tissues. The search for pathogenicity in these enzymes constituted thrust area in physiology of parasitism. Other areas where these enzymes have been studied are the wood and fabric technology, for cellulases and food industry for pectic enzymes. Interest in hemicellulases have been comparatively of recent origin. The plant cell wall is both chemically and structurally complex, containing cellulose, hemicellulose, including xylans, arabidins, and mannans, and pectin. Cellulose is a homopolymer of beta-1,4-linked dextroglucose. Cellulose microfibrils act as the structural backbone of the plant cell wall. During cellulose biosynthesis, chains of poly-beta-1,4-dextroglucose self-associate through hydrogen bonding and hydrophobic interactions to form cellulose microfibrils, which further self-associate to form larger fibrils. Cellulose microfibrils are somewhat irregular and contain regions of varying crystallinity. The degree of crystallinity of cellulose fibrils depends on how tightly ordered the hydrogen bonding is between its component cellulose chains. Areas with less ordered bonding and therefore more accessible glucose chains are referred to as amorphous regions. The relative crystallinity and fibril diameter are characteristic of the biological source of the cellulose. The irregularity of cellulose fibrils results in great variety of altered bond angles and steric effects which hinder enzymatic access and subsequent degradation. Because of this variability, cellulose degradation requires a variety of enzymes, presumably with wide variations in shape of the substrate binding pockets and active sites. Now let's talk about the basic cell wall material, that is cellulose. This basic structural or skeletal material of cell walls of all higher-land plants is also present in some seaweeds and is synthesized by a few fungi and bacteria. Cellulose is the most abundant natural polymer, and it has been estimated that 10 raised to the power 10 and 10 raised to the power 11 tons of cellulose are synthesized each year. The same amount is also destroyed, including that which is microbially degraded. Now let us talk about the cellulolytic enzymes, that is the cellulase complex. There are three types of enzymes in the cellulase complex. Number 1 is beta-glucosidases or cellobiases. These enzymes hydrolyze cellobiose and other cellodextrins. Also, other beta-linked glucose dimers and beta-glucosides such as para-nitrophenyl beta-glucoside yielding glucose. They do not act on cellulose. Number 2. Exo-beta-glucanases. These enzymes attack cellulose molecules from the non-reducing end. The best known ones are the cellobiohydrolases of trichoderma, which remove cellobiose units analogous to beta amylase. Exoenzymes removing glucose analogous to glucoamylase are apparently less common. Exoenzymes barely hydrolyze CMC, that is cellulomethoxyl complex, or other substituted cellulose derivatives because the substituents stop the action. Number 3, that's endo-beta glucanases. These enzymes attack the cellulose molecule in random fashion. They are usually measured on CMC by production of reducing groups or reduction in viscosity. Now let's talk about pectic substances. Pectic substances consist of closely associated polysaccharides, the major components of which are made up of a linear chain of dextrogalacturonic acids, in which varying proportion of the acid groups are present as methyl esters. The term pectic substances is commonly used for pectin, that's the methyl ester, pectic acid, that is deesterified pectin and its salts, pectates, for example sodium polypectate, and certain neutral polysaccharides lacking the galacturonan backbone, which are often found in association with pectin. Now let's talk about pectolytic enzymes. Enzymes which act on pectins have been studied in more detail than those acting on most polysaccharides other than starch and cellulose. This is because of their involvement in plant tissue maceration, which they cause by dissolving the middle lamella, that is the intercellular cement which holds the cells together and is chemically pure, exclusively pectic material. The other reason for interest in these enzymes is in processing of fruit and vegetable products. Enzymes acting on pectins are of 3 main types. First, pectin methyl esterase. Second, polygalacturinases. And third, pectin methyl transaminases and polygalacturonate transaminases. Enzymes of the first type hydrolyze methoxyl ester groups and presumably play a role in modifying pectin properties during growth of the cell wall. Pectin depolymerizing enzymes of the second and third types, and especially of the latter, are commonly elaborated by plant pathogens involved in the maceration and rotting of plant tissues, and in the decay of infected fruits and vegetables. Moving on, let's now talk about the xylans. Xylans are the most abundant non-cellulosic polysaccharides in majority of angiosperms, where they account for 20 to 30% of the dry weight of woody tissues. They are mainly secondary wall components, but in monocotyledons they are also found in primary cell walls. These are less common in gymnosperms, where the galactoglucomanans and glucomanans form the major hemicelluloses. Next is xylolytic enzymes. At present, 2 enzymes are known to be associated with the xylolytic activity of microorganisms, both of which are hydrolases. That's endoxylanase, which attacks 1,4-beta-dextroxylon at random, giving rise to xylo-oligosaccharides, and exoxylanase or beta-xylosidase, cleaving the same substrate from the non-reducing end, leading to the production of monosaccharide xylose. With that, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.